Hi, I'm Mark Rodonis, and I'm a biochemistry student at Arizona State University. This is a computational study of superoxide dismutase and ALS-causing mutants. Let's start with some background information. So what is ALS? Well, ALS stands for amyotrophic lateral sclerosis. And as we all know, it's a neurological disorder, as we're all familiar with Stephen Hawking. One of the things that you might not be familiar with is that individuals usually pass away within 3 to 5 years of the symptoms appearing, and only about 10% survive for 10 years or more, which makes it a very serious disease and one we're studying and trying to find a cure for or an answer for. If you look on the right, there's a comparison between a normal spinal neuron and a diseased neuron. As you can see, the fiber's been affected on the right side, and what this basically translates to is upper motor neurons, lower motor neurons degenerating, dying, and they stop sending messages to the muscles. The muscles eventually waste away and atrophy. And eventually the brain loses the ability to control voluntary movement and motor function, even down to the point where the individual is unable to breathe. So the most important part on this slide with regards to this presentation has to do with the 10% of the cases being inherited through a mutated gene. And we'll talk about more of this, but this is where we're going to look at is basically a mutation that is definitely associated with ALS. So there's about 2 different categories for ALS. 90% of them are what they're called sporadic, but 10% are familial, which means that they come from your parents. And of those familial cases, about 20% of them, 10 to 20%, are known to be SOD1 related mutations caused. That means that the ALS can be directly tied to this mutation in this enzyme, superoxide dismutase. And so it's been a large target of studies because of this. So let's take a look at the enzyme. If you look above, this is what a normal superoxide dismutase looks like. It's basically a homodimer. There's an interface in the middle there, and there's a beta barrel on both sides. This is a metalloenzyme, which means it coordinates metals. And in this case, it coordinates copper and zinc, which are involved in the reaction mechanism. If you look below, there's just a, each one of those little nodes, if you will, represents a possible ALS mutation in superoxide dismutase. And we'll talk more about how many mutations there are and different SOD variants. So let's talk a little bit about why mutant SOD, causes toxicity. So obviously it's responsible for getting rid of what's called superoxide, which is a radical that's formed through various reactions within the body. And if that radical isn't taken care of, obviously it can do some serious damage within your biological system. So down here, there's about 3 different reactions that are focused on. Peroxidation, which basically can lead to OH radicals, tyrosine nitration, and reverse catalysis where O2 goes back to superoxide radical. If we look at all the mutants here, this basically is an entire website dedicated to ALS SOD mutants. So there's over 100 different variants that are responsible or have been identified as causing ALS. And if you look on the left at the mnemonic, you'll see that most of them are just basically a single missense mutation, meaning just a changing of one amino acid residue to another amino acid residue. And so it brings about the idea that how can it be that you can mutate a single residue across this entire enzyme in so many different areas and still result in the same disease? There must be some kind of trend, something going on. But as of this moment, no one's been able to elucidate the reasons. So my project idea was to uncover a relationship. So the idea was to perform computational analysis on not just the SOD wild type, but also ALS-causing mutants, and just to try to gain a better understanding via thermodynamics and modeling to uncover correlations between behavior and properties of an enzyme. I used various tools involved in this project, and they're listed below: Gaussian, Avogadro, VMD, NAMD, QuickMD are tools that we all use in this course. I chose to use two extra tools, which were PyMol and Superpose as well. Okay, so what's the approach here? So because this is not just a simple enzyme, this is an enzyme that's a metalloenzyme, the metals basically require a little bit something extra. And what I mean by that is that when you run QuickMD with NAMD and VMD, you have to provide certain topologies. And those topologies are readily available for certain residues and certain atoms, but they're not readily available for metals. And so the approach is normally taken to overcome that when you want to model a system that has metals, is you generally will use larger scale molecular mechanics techniques for the bigger portion of the molecule in the system with the water and so forth, which is everything that represents outside of this box. And then you'll hone in on that specific area, whether it's the active site or maybe the active site with substrate, to use quantum mechanics or QM on that piece. And that'll usually require a tool like Gaussian, let's say. And then what you can do is you can systematically later on piece those two ideas together and model the system more accurately. However, it's a little bit outside of the scope of what this class covers. So what I chose to do was to basically begin working on each one of these pieces individually. And I'll show how I did that coming up. So let's look at basically the mutants that I chose. So the wild type that was chosen was PDB2C9V, crystallized structure. They're all crystallized structures. And there was 4 mutants that I chose to look at. And as you can see here, the first mutant was G37R, and this represents an early onset, relatively slow progression. It'll also be referred to as 1AZV in the PDBs. Then I chose H43R mutant, which is known for a rapid disease progression. Then G93A was chosen to be even more severe than was shown by H43R. Lastly, we're going to look at I113T, which is an SOD1-causing mutant, but now it's been bound to a 5-fluorouridine, which helps stabilize the homodimer structure. Now analysis on the molecular mechanics piece. So I like to start simple. So I used a program called Superpose to basically line up 2 different mutants, one AZV and one PTZ, just to see if there might be some identifiable differences in the structure. And as you can see at this level, it really doesn't, it's not very clear as to how they're different, or going to function different, I should say. So the next step was to load them into QuickMD and try to get an idea as to RMSD and so forth around these molecules to see how they behave comparatively to each other, because I thought that might be one method that we could use to understand the differences in the molecules. It's a method we learned in class, and so I figured we'd apply it. However, not so easy. Might be one of the more difficult steps of this process, because there were a lot of structure check issues with loading in PDBs, and some of them I was able to overcome and some I was not. That being said, topology and parameters would be an issue that I had discussed earlier as to why you would want to use a QM-MM approach, because you cannot load in certain topologies for metals. But a cis peptide bond is also an issue that comes up, and sequence gaps as well. So here I've highlighted the cis peptide bond, and when that comes up, you can load this tool, which basically you have to auto-generate a PSF and go here, and you can do some modifications, and it's supposed to correct the issue. However, I did not have success in doing so. So unfortunately, I had to leave out quite a bit of PDB structures just because I was unable to overcome this. That being said, this is what it looks like once I took care of topologies and parameters. So in this case, I identified 5, there's copper, zinc, and sulfates in there. And what I've done is you highlight them and you basically delete them. And once I did that, I was able to begin my QuickMD and NAMD analysis. So some of the settings that were involved, I had to change the type selection. I would remove the water-bound objects. I would also change the temperature to be more consistent with that of biological environments. So it starts at 27 Celsius. I move it to 37 Celsius. I use an implicit solvent, and the simulation time was 1 nanosecond. And I'll show why I chose that time in a minute. So here you can see two RMSD plots that come from this on about a 1 nanosecond time scale. And as you can see, one AZV and one PTZ, well, they don't look very different. And so this was part of a hypothesis I had because of the structures only being mutated really in one residue each of difference from each other. Generally didn't think there would be a huge difference in the RMSD, and this showed that. So I did try it on one more mutant, which was 4A7S, and while the time scale is not the same, once I got to about half a nanosecond, we decided to stop modeling this because it was obviously on the same trajectory as the other mutants, and therefore this was not going to probably show any kind of correlation that I was looking for. So therefore, we moved on to quantum mechanics computations. So in order to do this, I had to isolate the active site first, and that's shown here. This is done in a program called PyMol, and it helps to. I use it to basically label now the residues here for you to see easily as to what the active site looks like. And the active site residues are outlined on the left. They stay the same in all of the mutants, so that's really helpful. And so basically what was done, it was I selected these residues within a particular PDB that's loaded up in PyMol, invert the selection, and then delete everything else. And what would be left is what you're seeing on the screen right now. And this would be my this would be a tool that I was going to use in Avogadro. I mean a system that I was going to use in Avogadro eventually, and also Gaussian. So I want to just preface what I'm about to say next about some of the correlations I was going to find with the mechanism. So if we start with the top, which is 1, you might be, and obviously this is a little in depth, but the copper is coordinated to 3 histidines and the nitrogen off of the histidine that's coordinating the zinc as well. So there's a histidine between the zinc and the copper. And what ends up having to happen, if you look at number 2 where the arrow is that coordinated bond between that nitrogen and that copper will eventually have to break. And you can see that in the next green arrow on number 3. And so what's going to be shown next is that this distance between this copper and this nitrogen is what I looked at to analyze in each one of the mutants to see if I could find some kind of trend. Because I understood that eventually this copper has to be rebound to that nitrogen. And that idea involves a proximity to one another. So once again, I like to just outline that these are the mutants that I chose, and there is obviously an increasing severity in the actual ALS symptoms, with the exception of I113T, which we'll talk about at the end. But for the 3 first mutants, G37R, H43R, and G93A, they get progressively worse in their effects on the individual that has them. So here's the active site correlation. So what I did was I loaded these up inside Avogadro, and I was able to measure the distances between the coppers and the nitrogen on that near histidine that I said that was in between the zinc and the copper. And on the wild type, we end up with a 2.246 angstrom distance. So we'll use that as a baseline. Now, once we go to the early onset, relatively slow progression mutant, G37R, we see that this distance now between the copper and the nitrogen grows to 2.647 angstroms. Once again, starting from originally 2.2. But here's what happens when we get to a rapid disease progressing mutant H43R. Now the model's not even loading up with the copper and the nitrogen coordinated to one another. So we've now reached a 3.151 angstrom distance, and the correlation is starting to show something where this distance is starting to become related to the actual disease progression. So I was kind of excited to see what's going to happen on the most progressive disease mutant, which we'll show next. Actually, before I did that, I had to actually bond the previous H43R, and I drew a bond between copper and nitrogen because this is going to be required in order to actually run this through Gaussian, which I've shown here. And last but not least, G93A. It's the most severe progression of the variants I chose. And as you can see here, it has a bond. I've added this bond here. But the distance between them is 3.579 angstroms. And once again, I've added this bond manually in order to run this through Gaussian, which was required. So after I loaded into Avogadro, I had to actually build and add hydrogen atoms to it. You couldn't run it in Gaussian without doing that. And there were some adjustments I had to make as well to the configuration files to get it to work. But once I did so, I was able to get some results. And here's what I found. So for the wild type 2C9V, which is kind of our baseline, we had a negative 6,600 atomic units, Hartree's energy. Then when we get a little bit further from that nitrogen, the copper from that nitrogen, we end up with a higher energy, negative 6183. And then as we get even further to the more rapid disease progressing H43R, we come back down to an energy that's close to the original wild type. And what this represented to me was almost as if there is kind of a maximum of instability between that distance where H43R is, as far as from the copper from the nitrogen, and where the copper is the distance that it is in the normal wild type. And so what may be happening in the G37R variant is that copper may be kind of rocking back and forth between there. And that's why it may be able to have some function, but not enough to actually not cause a disease. And last but not least, once again, the most severe progressing version, a variant of the mutations is G93A, and that one had the lowest energy of the sorts. So now I said, well, maybe I can apply this knowledge. So what I did was I took a known SOD1 mutant, I113T, that causes ALS, and there was a paper written up about 5-fluorouridine being bound to it and actually stabilizing the homodimer. However, the homodimer and stabilization and the copper to nitrogen distance seem to be correlated, even though those two sites are very far from one another, as you saw earlier when I showed the actual homodimer structure, where the coordination of the metals is not anywhere near the interface between the two dimers, between the dimer complex. So therefore, if you look at the distance here, funny enough, even though this is a mutant, the copper and the nitrogen are closer together than they are even in the wild type. So this might give us a little bit of insight into how to possibly develop drugs to make the enzymes more stable within the organism and stop these aggregations that happen in ALS individuals. So summary, the next logical step. So I would say next steps, if I were to continue working on this, would be to figure out how to correct the cis peptide and sequence gap issues, so I can apply analysis to more PDBs, perform an actual QM analysis as opposed to breaking them apart and not tying them back together at the end, and to bind a substrate maybe to the active site and try running Gaussian on wild type and mutants to understand the energy differences there. Where can this lead us? What can help us maybe to understand how to build better drugs against FALS, as I had already mentioned. Maybe a correlation between the stabilization of the homodimer and the active site might help to understand the bigger picture, which might explain how many mutations can cause the same disease. And ultimately helping to design gene therapy studies. Maybe that might help redesign the faulty enzyme in vivo eventually down the road. These are my references. And thank you very much for your time.